Okay, how's everybody doing today?
Good.
Right. So my name is Rohan, and today I'll be presenting my talk, "Fighting the Flu with Machine Learning." So, the up there is my Twitter handle and my GitHub. If you didn't catch that, it'll be on the last slide. So when I'm taking questions, you can look at it. So I am going to be an 11th grader this fall in high school. Thanks. Thanks. So I've been programming for 2 years now, mostly in Python. I've also done a little bit of Java and R, and I'm not very proud of the Java part because I did start with Java, and that gave me a terrible introduction to programming. So yeah, after I started Python, I was like, this is way cooler. So I'm doing Python now, and specifically, I like scientific programming and data analytics and machine learning, stuff like that. So today I'll be and I like doing projects. So today I'll be talking about one such project. So let me start with the problem. Is the flu. So chances are you may or may not have had the flu before. So millions are affected worldwide, and there isn't a great way for scientists to make vaccines for the flu. So they just vote on what drugs and antibodies should go into the flu vaccine for that year, and this practice has been called questionable by the National Institutes of Medicine. So, and sometimes the vaccines don't always work. If you remember, in 2009, there was a worldwide pandemic of the H1N1 flu virus. So what I wanted to do in this project was create a better way for scientists to come up with vaccines, and my solution to that was to come up to predict how flu strains would look like in the coming year so that scientists could analyze that and then have specific vaccines tailored towards those specific flu strains so you'd have a lower probability of getting sick. So here is a nice graphic of how the flu infects a host. So in that first panel, you've got the virus, which is the smaller circle, and the cell. So those little purple dots on the outside of the virus, those are proteins, and they attach to the receptors on the host cell. So, and then after the virus gets in the cell, it dies and it injects its viral DNA and RNA and its other genetic material. And as the cell grows, so do that DNA, RNA, and those proteins. And eventually, when the cell is about to die, all those viruses leave the cell and they infect, and they go on to infect other cells. So, from this research, I decided to look at those specific proteins, those little purple dots on the outside of the cell. In the flu, those are called hemagglutinin and neuraminidase, and being biologists, they had to come up with extra long names for them. So for now, we'll just refer to them as HA and NA. So HA regulates whether the virus can enter the cell or not, and NA regulates whether the virus can leave the cell. So I thought to myself, if I could knock those proteins out or come up with or predict how they would look in the future, then scientists can create vaccines which could take out those proteins and thus prohibit the virus from entering the cell. So this is my approach. So I would first predict future genetic sequences of the flu, and scientists could analyze that, and I would do that using phylogenetics and machine learning. So phylogenetics is a study of the relationships between individuals on an evolutionary tree. So this is pretty much a phylogenetic tree. You've probably seen one of these. So right there on the bottom you have E, which is the child of F. And so what I would do is I would say, how has the genetic sequence of E changed from F, and how the genetic sequence of F has changed from G. So if I could train a machine learning model to analyze these relationships as the flu changes generation by generation, then maybe I could predict like maybe a child of E. a child of E, maybe what would that look like? Maybe would it be AACC? Would it be AATC? So I wanted to figure that out. So my setup was to first obtain data from the Influenza Research Database, or IRDB, which is funded by the National Institutes of Health, and read in the genetic sequences of hemagglutinin and neuraminidase and train a machine learning model based on the phylogenetic relationships. So what I wanted to do was train the relationships between G, F, and E. So how has F changed from G and how has E changed from F? But me being inexperienced as I am, I wasn't sure how a homemade machine learning model would work because the flu data is quite complex, and I wasn't sure if my homemade algorithm could match that level of complexity and predict as well. I also wasn't sure what machine learning model to use, how to measure accuracy, and how to read in a FASTA file, which is a common file format for biological data. So this is where my first library came to my rescue, and that was BioPython. So BioPython deals with everything biology in Python. So I used this library to get past that initial hurdle of how to read in a FASTA file. Originally, I was reading it in as a regular TXT, and that wasn't working out very well because when you read it in as a TXT, there are a bunch of like random characters that are in a FASTA file which show up. So what I used to do was look for patterns, look for specific characters which show up right before the gene sequence actually shows up, and that wasn't working out very well. So with BioPython, I was able to read in a FASTA file much more easily. So after I got past that hurdle, it was time to create the algorithm myself. So I did some research and I found a decision tree algorithm would be the best fit because it was easy for me to write and it would be easy for me to debug, and as well it matches a phylogenetic tree, as every time a decision tree branches, it's similar to how a phylogenetic tree branches from its parent to the children, and as a decision tree branches from one question to more questions or a decision. But there were lots of problems. So I wrote a classification algorithm, although what I really wanted was a regression algorithm, as the algorithm was too simple and it underfit the data. So my initial hypothesis was confirmed because I could not write an algorithm that matched the level of complexity that the flu data had. So scikit-learn helped me. As most of you know, scikit-learn is the go-to machine learning library for Python. And with this, I was able to use lots of algorithms. I could just import them really easily. I didn't have to write them myself, so that saved a lot of time. So now with BioPython and scikit-learn by my side, this is my setup, which is updated to reflect the changes that I made. So the first step stays the same. I still get the data from the Influenza Research Database. Next, I would read in the flu genetic sequences using BioPython, and I'll show you how easy it was for me to get that data using BioPython. And I had to add another step, which is encoding the data. So with scikit-learn, or rather with my previous algorithm, I would simply input the genetic sequence, which is a big string, like roughly 1,700 letters long, ATGC, just random combinations of those 4 letters, and I could just input that directly into my flu prediction algorithm. However, with scikit-learn, it only takes numerical input, so I had to encode those letters into numbers. So I had to add that step, and the last step stayed pretty much the same, with the exception that instead of writing my own algorithm, I would use the scikit-learn library. So this is a diagram that I drew showing you the overview of everything I would do. So what I would do was create a phylogenetic tree for each protein. So pretty much the grandparent to parent to child relationships between between flu strains. Next, I would encode the data, and then after that, I would give the data to my scikit-learn algorithm and test the accuracy using cross-validation, and then add more data and just continue doing this until I reached a favorable result. So this is what I could do with BioPython. In just one line, I could get the data which is stored in that in HA100, which is 100 FASTA files. And by doing that putting them into a list, I had one big list of all the different FASTA files. So what it would be a grandparent, a parent, and a child, and then a group of 3, and then another grandparent, parent, child, and so on for 100 of these. So instead of looking for specific characters which signal the beginning of a genetic sequence, I could write in one line, and I could have all of the relationships ready. So this was my encoding method. So essentially what I did was, for A corresponded to 1, T corresponded to 2, and so on. So what I would do was I would take these huge strings of A, T, G's, and C's and convert them into one big number. So what I did initially was take all of these letters and put them in one big int of 1700 digits. And I soon found out that only 5 of them were being stored. So I tried putting them in a float. That worked a bit better. However, it could only store up to 15 digits. So what I ended up doing was breaking them up into floats of 15, and then putting each float of 15 digits into one big list with roughly 80 elements. So I would have 1, 2, 3, 4, 5, 1, 2, 3, 4, 1, 2, 3, 4, up to 15. Then another element in the list with more digits, and so on for roughly 80 elements. And that would be the entire genetic sequence of the HA protein. And this was my decision tree algorithm before scikit-learn, and this was my decision tree algorithm after. So this is not even the whole thing. This is like half of it. So pretty much from 80 something lines to 6. So that was incredible. I was blown away. Like for me, I've never seen this before. So I was really excited, and it turns out this had really good accuracy as well. But there were some drawbacks in using BioPython and scikit-learn. BioPython not as much, but with scikit-learn, I had to stick with training my machine learning model on simply the parent-to-child relationships, because in scikit-learn you don't have that. I couldn't add that extra generation of that grandparent, which I would like to add, because more data always results in a better model. You can't beat more data. So I could only go one correlation. So I could only go from parent to child. And scikit-learn also couldn't take letters or characters. And I wish that I could have put all of those base pairs, those ATGC, into one big int. So I wish it could hold more significant digits, but you can only get so much. So now I'll talk about my results. So this is H1N1. So H1N1 is a subtype of the flu. There are lots of different subtypes. So I trained mine on the most common ones which infect humans, because I want to protect humans. So so H1N1 is this was the one that broke out in 2009. It caused a global pandemic, resulting in a lot of deaths. It was actually the most potent flu since the Spanish flu all the way back in 1918. So this one is especially dangerous because it mutates very often. And although this does not affect humans that much, thankfully, but it still mutates really quickly, and that makes it really dangerous. And this is another flu subtype called H3N2. This is more common in humans and doesn't mutate as quickly. So you probably noticed that the accuracies with these three three algorithms, decision trees, random forests, and extra trees, they performed a lot better in H3N2. And I think this is because H3N2 doesn't mutate as much, so it's easier for the algorithm to pick up on those changes because there's not as many. So this is H3N2 and this is H1N1. And those two bars, the yellow is the neuraminidase, which regulates the exit, and the blue bar is the hemagglutinin, which regulates the entry of the cell. So with the exception of the decision tree algorithm and extra tree algorithm, my model performed really well. So I ended up going with the random forest algorithm. And these three are all decision tree based. So I compared my algorithm with two previous European studies. So this one right here is a German study, and this one is a study done in Spain, I believe. So both of these do not use machine learning. Rather, they just rely on formulas to predict how the flu will change in the coming years. And as you can see, the results look very encouraging. So to sum it all up, I wanted to create a better way for scientists to make vaccines for the flu, so how I went about doing that was to predict how the flu would change in the coming years, so that scientists could create vaccines specifically tailored towards them. I used BioPython and scikit-learn to make my algorithm shorter, as you saw in the how scikit-learn helped me, and the results look very encouraging. So thank you all very much. So yeah, I just feel I'm taking questions right now. So anybody? Yeah. Yeah. Mm-hmm. Yes, they code for the amino acids, but what I decided to do was, if you can take out the sequences that are coding for those amino acids, like take out, like if you can create a antibody which can perhaps change one of those sequences, like I'm diving really deep into biology here, but so I believe UTA is the codon, which is that three base pair long sequence to stop coding for any more proteins. So if you can maybe create an antibody which would change, which would perhaps move that little codon in the beginning, then you can stop that hemagglutinin from being coded, so it won't be able to enter the cell. So I was just predicting that. Anybody? Oh yeah, sure.
Oh yeah, congratulations. That was a great talk. Were you able to communicate your results to any researcher?
No, I haven't done that yet. That's definitely, I'm gonna do that this summer, because this was for a science fair in my community. So after that, I'm a high schooler, so it's lots of APs coming up. But now it's summer. So I think that's definitely on my to-do list before school starts, is communicate my results to scientists. Definitely sending out some emails. Yeah.
You said you were encoding a set of base pairs as numbers and storing them as long floats in a list, right? So why not store them each as codons instead of a set of 15?
That's actually a good question. Like I could actually do it that way. I think I'll definitely try storing them as individual codons, so that scientists can like see where each one is occurring. Like, how do I? Yeah, I mean, I could definitely try it like that. Any other questions? Yeah, all right. Thank you very much.